Hello, everyone. Many thanks for joining us today. I'm super happy to have Professor Ville Pimenoff here today with us. He's a group leader and head of Exposomic lab at the University of Oulu, Finland, and he's actually investigating biotic and abiotic life course exposures in health and diseases with EU Horizon Project and Finnish Cancer Foundation fundings. His group has been particularly developing wearable devices assisted bioinformatic methodologies for longitudinal aerosols monitoring of chemical and microbial exposures. He's also a visiting scholar in Stanford University and a scientific board member in the European Human Exposome Network and the International Human Exposome Network. I have to say, he had recently a surgery, and despite everything, he agreed to be here with us today. Many thanks, and the floor is yours.
Thank you, Patricia. And yes, indeed, it's a winter season, and I heard myself on the slope, but luckily in a shape that I can give you the talk. Uh, as you just heard, so I thought on giving you a lecture on two main topics, which is in my lab, which is estimating biotic, so biological and chemical environmental exposures, either at individual or community level, which requires different approaches. And let's see if we can get the system running. Yes. So I believe most of us are familiar with the concept of human exposome, but as in the definition, my work relates directly into the core of it, which is trying to understand the life course exposures, which are basically defined as all the different exposures that we get in our lifespan. Here's a sort of a listing of different components of the human exposome, either from the ecosystem level, going into lifestyle, social, physical, and chemical aspects. My talk will focus mostly on the outdoor and indoor air, which is the concept of more personalized exposure analysis, and then on wastewater surveillance or wastewater sampling to use as a community level exposure estimations. So these are two niches in the comprehensive concept of human exposome. And the initial question to go on this research is that we were aiming to monitor environmental exposures that might affect health, and in this case, for instance, on uh, female pregnancies are known to be exposed by several factors that cause adverse pregnancy outcomes, but there's a very large unknown component in several of these adverse outcomes, and we thought that we might be able to capture some of these signals by doing a proof of concept of human exposome, studying 100 pregnant, non-pregnant Finnish women by monitoring them at least 4 months during their pregnancies. And in this particular project, we ended up using ultrasonic personal air sampler, which is a wearable device, which is one of the unique ones in the sense that although it's an air pump with a filter cartridge that you change the filter that you use to capture all the particles from the air. You can very well control it by mobile phone to normalize the airflow, and by using this kind of a shoulder strap, um, You can wear it 24/7. And in this case, as we gave that to pregnant women, they were receiving it very well. Even though it's very intense sampling measure, they were ready to wear it 24/7. Only when sleeping, they put it on a table in their bedrooms. And what this wearable device allows, there are several versions of it. The ones we used, it's from the filter. We can do very deep resolution biological profiling by DNA, RNA, metagenomics, and then chemical profiling. In this case, we used non-targeted mass spectrometry. And also, the device has its own GPS coordinate recording. So in the sense, we can control all of these sampling time points with the exact location or routes of at least the device, and in this case, as a proxy of the person who used the wearable. And here's our sampling scheme for 2 months on the summer season and 2 months on the winter season was the time frame that we wanted to have so that we can also capture the seasonality of environmental exposures. And the main sampling methodology was using the UPAS personal aerosol sampler, but we also collected urine, blood, saliva, and monitored the cardiac rate during these monitoring periods. And in that sense, the women actually took it very well on even in during their pregnancies to have this type of monitoring because they were very interested on knowing their environmental exposure patterns, especially if it might relate to their health in any way. And at the end, we managed to have the 100 women recruited, and then the data out of the variables is we used Ultima sequencing, is one of the newest sequencing methodologies available through our collaboration, and then mass spec on the filters, but also from the urine samples of the individuals. Here's sort of a schematic diagram on what the current UPAS devices are capable of. So you have the GPS coordinate temperature, humidity, and PM measures as their own sensors, but then you basically divide the filter into nucleic acid extraction and further into sequencing, and then chemical extraction, and then mass spec analysis, depending on your interest and methodology. Obviously, normalizing is very important in this type of longitudinal data. So we used automated library preparation and Ultima sequencing because it is a method which gives a longer sequence reads than standard Illumina methodology, which we at least believe that gives a better and more robust results as an outcome. Here's an example of what type of data we had altogether. Here's a phylogeny of all the biological material found in one individual. This is from a few years follow-up. This is the previous data published on similar device. But then also that we had the GPS coordinate on also to estimating mobility of these individuals and the cardiac monitoring in very high frequency. And now on focusing on the biotic exposure and what are the interesting parts of it and the challenges we noticed. So when looking on the pathogens related to human health, we know that most of these microbes they evolve interaction with the host population. And important note here is that pathogenicity is mostly strain specific. So any microbe the strain information is important. However, especially when you're using environmental sampling, the huge challenge is how to be able to detect the strain or even the species. And as an example, here is a well-known viral family affecting humans, but which is representing more than 200 strains, but only 15 are sort of pathogenic. So in we are dealing with a high diverse a window of biological matter, and the challenge is when getting the metagenome DNA RNA based metagenome from the wearable device, how to comprehensively capture uh, not only the bacteria, fungi, and the likely at least human genome, but then, for instance, phages, other viruses, because their genome size varies. But then, of course, in environmental exposures, the most of the biomass is very low, and in that sense, it brings a challenge on how to reliably detect. And as an example, here is a few years back a famous paper on just looking on the microbiome. On this was in clinical samples, but with the fast track classification methods, where in the left on the red they detected a huge amount of different microbial taxa, and on the blue you can see those taxa that were actually confirmed that existed in the data. So the bioinformatic pipeline methodology is very crucial here in the sense that even in human tissue related samples, there can be a huge biases on what you capture from the metagenome. But then when you go into environmental sample, like in the wearable device, it is even more challenging to make sure that you have a solid detection of the biotic exposures. And here's a sort of a quick schematic diagram on our methodologies, which is basically taking all the metagenome data into direct taxonomic mapping at the first stage, but also doing de novo assembly and then merging all that information together and doing a deep quality control that we can be sure that what we detect, at least on the species level, is truly in the data, and it's not due to the mismatch or a low quality of parts of the metagenome data. And here's an example of that type of validation where uh, the plotting shows the uh, abundance of each taxa. So every dot is a taxa classified in the samples. And then there's a breach of coverage. So how big part of the target genome was detected with no mismatch distribution with the reference genome used in the full classification of the data. And basically what it shows that the blue and the gray taxa, those are the ones which are mostly reliable, and especially the red and then orange ones are very likely very noisy signals or truly false positives. So that part is very crucial in the environmental exposure analysis, and after curation of the data, here's an example of one sample so same sample and DNA RNA analyzed. So with our classification pipeline, we could classify most of the reads or contigs of the metagenome, and also that depending on the RNA, those will capture a bit different parts of the biotic. But for instance, on viruses, which often are a very small fraction of the biomaterial, we could detect them even in very low abundance. And another way of showing the challenge here is currently there are several methodologies to detect different microbe taxa. Here's one typical, which is a Kraken 2, that do not validate much of the classification. It finds the closest phylogenetic match of the reference genome, and here the other is a method we used in most of the environmental microbe classification. So there are quite a few which we classified as false positive on, but found on the Kraken. And on the other hand, we found several which could not be found in these standard methodologies. This, I would say that it's a huge issue, but it's an issue that you need to be aware when using different type of detection pipelines, and also on what type of references you're using. So these are all reference-based classifications. And here's a bit of a summary on example of the same data sets, but used on Kraken 2 and other, which means our current pipeline, that you can detect quite a different fraction of the metagenome in the environmental samples. And that again brings the question of like where you draw a line on true positive detection. So here's the curated examples of fraction of the UPass data. So the main domains found, we can see that bacteria taxa were dominating. And on the other hand, this is just a snapshot of across the samples, either from the same individual or different individuals. And interesting here is that on the heat map on the right-hand side, on species level, we already see very a clear pattern of change in the biological exposures. Oops. And then when we dive into the different domains, if you look on the bacteria, we can see like a very broad taxa classification of almost 6,000. But then after curation, there's about 200 abundant good candidate taxa that shows a clear seasonal variation. So the comprehensive diversity of the bacterial taxa is a lot larger in winter months than in summer months. And thinking of the individuals wearing the wearable device in winter months, you typically in places like in Finland, you are indoors with a more dense, so people are closer to each other, and indoor air. So that's what we already see as an indication that there's a clear difference in air regarding summer and winter months. Uh, then, when we look in more detail of seasonality on the right hand side graph, you see that there's a red bars in very low abundance, which is interesting that there's clearly a plethora of Bacterotaxa which are more present on summer months. These are the major patterns that can be already seen in this kind of a shallow level analysis. Then when we look on the same for the viruses, we can see that there's a uh, quite a huge cloud of viral traces, but most of them are very tricky, or I would say that not well classifiable, either because of the actual read quality or content quality. And but however, on using those abundant viral taxa, we could see that their diversity pattern is very much larger in winter months than in summer months, which is again a typical time when viruses are spreading in the population, and also virus in this case we could detect several bacteriophages, which is another interesting feature of the data. However, that needs to be looked in as a separate, because those detection of phages is not straightforward from metagenome. And then going for fungi, again, similar pattern, that there's a large collection of classified taxa, but abundant, clear classified taxa are only about 50. And interestingly, when we look on the seasonality, even on the shallow diversity pattern, we see that there's no clear pattern between summer and winter months, which is interesting compared to what we already see on the viruses and bacteria. And however, there are, for instance, effect size difference as seen on the right-hand side, and there are certain taxa which are clearly more abundant in the summer than in the winter months, and vice versa. But these are also only on a fraction of samples, and obviously it affects a lot on what cofactors you adjust on this data. And also an interesting factor that there was also intervertebrate exposures that we could find, and that tells what is the challenge of the environmental exposure when you look on the biotic fraction, is that you have all the living matter, and then depending on what uh, is run through the air pump for the filter, then you will have a very diverse biological cloud to be classified. And in that sense, it needs a lot of curation to be sure that what are the true positives, and what might be traces or misclassifications. Then going a bit on the, let's say, individual classification. So the dots are different individuals in our monitoring cohort. And here, when we merging together all the microbe cloud, so bacteria, virus, fungi, and so forth, interesting is that without putting any metadata factors, we can clearly see certain clusters, and those are individuals who are from same neighborhood, so same community. Uh, Clear individual level of variation exists as well, but we clearly could see that there's a uh, depending on the community, there were certain correlations which we could already see in the networks of the biotic exposures. And on the other hand, here's an example of the whole data set looking on the biotic exposure on the left-hand side, winter, summer months. We see that there's a strong diversity pattern, but when you merge all the microbes detected together, then the patterns get masked. And on the other hand, when we look on the right-hand side, is one of the plots of the chemical exposures, and which is plotted with the time of sampling, and we can see that the chemical exposures do not show strong variation, at least on seasonality on at this level. So these are two very different and very interesting patterns that we already saw on at this level of the work, which is now ongoing. And then I want to jump into more into the community level detection, and as an example here, using wastewater samples that we had in from 15 communities collected in Sweden during the COVID pandemic, and we did the microbe and the chemical detection from those wastewater samples to be able to trace on what we can predict as an exposure from the wastewater to the community. And here's the list of results on viral lineages found on the waste or full wastewater data. So 25 viral lineages, viral species, which are either infecting humans or infecting bacteria that are typical in human body, and also bacteria which relate to human gut microbiota, or otherwise are considered typical opportunistic pathogenic bacteria for humans. So these are the biotic uh, exposures that we could find from the wastewater data. Um, on the left-hand side, you see the main domains. So when you look on the full biomaterial, you see that bacteria are dominating on the wastewater, but we could get even a very nice fraction of the viruses, which is amazing, just using on metagenomics. Targeted sequencing would be more accurate here but because the wastewater samples, how they were collected in a controlled flow of the water, so we can also compare the results between the samples quite nicely. And what we already can see on just looking on the domain level variation here on the right hand side is a sliding window of archaea on the red, and viruses on blue, and on the sliding window of 4 weeks between two communities. On left hand side is one, and on the right hand side on another one, and there's a very strong fold difference of the viral abundance between those two communities. And I would say that it's amazing how you can measure that on just from wastewater. Obviously, then the next question is what are behind these viral detections? When we spread then on the analysis to look on three communities here as an example, either all the viral species, the bacteria, or the chemicals, we could clearly get the pattern of a significant different either viral, bacterial, chemical exposures in three communities. And interesting here is that it's the frame of the difference is rather similar when you look in more detail. And here basically visualized by how the ordination brings these three communities as a separate. And going in more detail of the data, on the left-hand side, starting with the human adenovirus with strain 41, uh, typical in causing infections in humans. We see that there's between these two communities, there's a clear um, pattern of outbreak, which interestingly coincidence exactly on the same time when the COVID pandemic was ending then and the social restrictions were released. So this is something which might be related. Obviously, on that would need also, for instance, a COVID detection. But in this data, we did not have RNA analysis done. Then in the middle, we have phages infecting Faecalibacterium species, typical in humans. Again, we see there's a clear significant fluctuation of these phages in the wastewater. And then on right-hand side, Clostridium, Salmonella enterica, typically pathogenic in humans. We see a clear fingerprint on these two communities. So even on let's say rather few time points, you can have a quite good detailed view on what might be the exposure of a microbe, in this case, for instance, virus, causing local outbreak. And for instance, the adenovirus outbreak that has been confirmed with other studies also on the same time in Sweden, then these samples were collected. So that is very interesting finding. And finally, then on when we're looking on chemical and biological exposures, another layer of analysis is that we look on the correlations of these chemical and biological exposures. Here on the left-hand side, you see a heat map of all significantly correlating um, chemicals to the 32, and in the end, it's 16 human-related microbes, either viruses or bacteria, mostly. And already thinking that this is the data from wastewater, um, I think it's a remarkable that with a metagenomic methodology, we can see several correlations on biological and chemical matters that are related to human presence. Obviously, then the next step is to try to figure out on what are the correlations that have some level of causality or relatedness on human presence, or for instance on the chemicals affecting on the biomatter. And here, as an example, then sliding windows of positive correlations to the biological exposures. Here are all the positively correlated chemicals, and then in as a sliding window in two communities. So we see that the resolution varies a lot between the chemicals. Then when we have negative correlation, there are a few very strong signals of chemicals having negative correlation. And then as an example to the adenovirus outbreak which we could find on the viral detection, then all those chemicals that correlate with this outbreak pattern. So this kind of an integrated data allows a very dense resolution to understand what is the likely exposure pattern in this case in the community and the time span that you have sampled those wastewater samples. And of course, here is then the tough question and the challenge that if you look at in more comprehensive way, here are the two communities that we had the samples collected between 2021 and 24, and here are the chemical patterns that when you increase on only having 140, we detected thousands of chemicals and large fraction of chemicals that cannot be classified either. So we see that there's a certain common patterns, but how do you control what is the best way of normalizing this type of data into the estimation of exposure? That is a challenging question. But on the other hand, the current methodologies allows uh, to create and curate these very large and diverse data sets, which then allows a lot of inference. And last but not least, on an example that I wanted to also include in this presentation is when we talk about exposures and how we could control and try to relate it to the full landscape of environmental exposures. In the pregnant women cohort using the wearable devices, we had the GPS coordinates also from the devices. So we built a platform to try to estimate also a different way. So GPS coordinate based exposures of those individuals. And here's a map of urban areas in Finland. So those are all blue areas. And this is land cover data. So in practice, if we look on the full Europe, Finland is mostly rural, and that's important here because then based on the GPS coordinates that we're recording every second, here's an example trace of the data, and we build an algorithm that could classify when the device is in the urban area and when it's not in the urban area. So urban areas are marked red, and those are all those communities that the individual has been visiting with a device in a certain time span. And using this GPS dense information, we estimated the cumulative time spent either in urban or rural area. And when we looked the correlation between this cumulative time either in rural or urban area with the biotic exposure, interestingly, just without controlling too many cofactors, we could find very interesting correlations between rural and urban, even though that those are rather shallow differences in the environment in Finnish perspective. But then on going in broader, thinking larger metropolitan areas, Stockholm, Paris, so forth in Europe, these patterns will likely be even stronger. But in the pilot work, we got very excited to be able to actually see certain pattern differences here regarding urban and rural area. And this stems from the question that originally the 100 women were recruited to half of them live in the rural area and half of them in the urban area. But we noticed that without integrating the mobility, the estimation of the rural or urban exposures can be quite masked, and therefore you would benefit a lot from the GPS information. So to summarize all this about human abiotic and abiotic environmental exposure, so in my opinion, to truly estimate what are the exposures that might environmental exposure that might affect your health, you need a well curated longitudinal sampling, and wearables are very good in providing this. On the other hand, any type of omics data is complex, sparse, and highly variable, and especially environmental omics data. So validating it and harmonizing is very crucial on the interpretation. Already at this stage of analysis, we can see that this personalized monitoring using variables gives a very interesting dynamic perspective of the exposure patterns at individual level, and then we see certain similarities in at community level, but this needs a little bit more work. And last, the wastewater sampling. It is typically used as a monitoring of particular pathogens, for instance, example as a COVID. But I believe in that well-curated sampling allows a very good estimation of the community level exposures, including chemical, but particularly biological outbreaks that can be very much on the public health interest. So here's the thank on my collaborators in Stanford, especially on give me advice and help on the methodologies on the wearable device details and data creation, and then the Supercomputing Center in Finland and all the study nurses and collaborators in these projects. Thank you very much.
Thank you. This was a really extraordinary and great talk. Any questions? So
So
I'm
I can see the chat.
There are no questions there. So I'm going to start, and if other people have a question, they can chime in. For the variable that you had for the pregnant women, was it only for environmental factors, or could you also, like, for the pregnancy itself, collect any data regarding the status of the pregnancy or any complications resulted through pregnancy?
That's a very good question. What I didn't say here on the presentation is that um, we actually collected information about the pregnancy. We also got information about the new offspring, and this is a ongoing work on the cohort. But yes, it's on one point is that we only gave the the wearables to the women after 2 months of their pregnancies for ethical reasons. But then, yes, the main interest in this pilot already is to see that what we can relate on any of the complications that we know of some of the women already had. Um, I can say that some of signals patterns are present, but we still need to do work on that to be more specific.
And another question on the same pattern, on the same context. So, when you are looking at pregnant women, um, because they are, so let me put it this way, because of like the whatever we are reading, is it from the women itself, or can we also like differentiate between what we are reading from the women itself or the baby infant?
That would require a separate sampling. And yeah, we have a part of the cohort samplings not analyzed yet, but already on the proxy of for instance, the women's health during their pregnancy. We have all the clinical information as well. And in that sense, we can at least correlate that. Uh, of course, uh, from the wearable data, it is data collected in every 2 weeks time point, so it's a cumulative. And that makes it a little bit different than if you have, for instance, uh, let's say frequent blood testing on inflammatory markers and so forth. This is a work which we haven't finished yet, but yes, we have linked the health data to the device information. But it's limited with these 2 weeks time points. That's the tricky part.
Thanks. Any question from the audience?
I have a question, if I may. Um, One of the challenges with exposomics is there's so much to measure, right? At so many
Yeah.
different time points across so many different areas. And I guess my question for you is sort of a bigger picture perspective on this. What is your sense of what you would really love to have? You just, as you just said, you're getting the chemical exposure over 2 week time periods. By the nature of it, we have to sample. Um, do you, in your perfect world, are you more interested in getting more and more people, or more and more metrics for the same people on finer time points? What do you think is going to get us? Because these are complex, really complex patterns with lots of variables. And so the answer is maybe everything. But where do you think we have the most room for growth?
That is a very good question, and thinking which I think is much needed on the whole exposomics field at the moment. Uh, it is, I wouldn't say dominated, but a lot of research is in chemical exposures. Uh, Particularly on your question, I'm interested and curious on more on how do I say it, the healthy exposure. So typically we look on exposures that are harmful for us, but what I'm also interested on especially using wearables, is that how we can capture and know a bit more about what are the sort of healthy exposures that we have, and then having a personalized device that you could get a robust measure of this would be very interesting compared to this type of a research-oriented, very dense and expensive work. So, and I don't think that we are yet there that we can comprehensively measure the major components of the human exposome. Um, it still needs a technical innovation, I think, to bring it into, let's say, lifelong or very much longitudinal collection of measure points. But it's difficult to say is that what should we measure, what would be relevant, and in that sense, my answer is that not only on looking on what is harmful for us, but also what is somehow health preservation exposures.
Yeah, thank you.
Any more questions? Thank you, Chrissy.
Can I ask a little bit of a silly one? You showed the invertebrate data that you got on your
Yeah.
sampling, your omics sampling, and I saw C. elegans there, I saw oysters there. Do you think that those were actual exposures that people had? I mean, it's entirely possible, right, that someone was exposed, that was working in C. elegans lab, for example, or was this or was eating oysters or something. It's just it was a really good metric of how messy this data can be, but I'm also wondering if it actually is just real.
That's a good point, and that's why I show several slides on that. Curating the data is very important. Uh, for us, some of the signals that we find is very puzzling. Is that, yeah, it's possible. Um, Also, a detail that women did not always wear the device in their shoulder. Some of them said that, yes, sometimes I put it in my handbag. And so thinking of what is the source and where,
Mhm.
uh, like what type of air and particles got into the filter and why, uh, is is very interesting, and all those classified tasks that I showed, those are the ones that we definitely can find from the data. Then the question is, as you addressed, is that um, why is it there? Why do we detect it? And that's another one which is for the sampling and the methodology, that it's not straightforward in all aspects. But that's why we have a quite detailed information of every individual on their livelihood and environment. But some of the individuals traveled as well quite a lot, and yeah, it's interesting that what you can pull out from the filters.
Yeah, thank you. I thought you did a really good job explaining sort of the messiness of the data, and I appreciate that. It's just really fascinating what you can find in environmental DNA assays.
Yeah.
So thank you again, and many thanks that despite the surgery that you had, you agreed to still give the talk. I'm super grateful, and I wish everyone a happy and nice rest of the day. And again, many thanks, Will.
Thank you, Patricia.
Thank you. Bye.
Thank you. Bye.